Hi everyone, and welcome to the Beginner Bioinformatics for Parkinson's Disease Genetics course from the Aligning Science Across Parkinson's and the Global Parkinson's Genetics Program. My name is Makarious, and I'm a trainee at the Laboratory of Neurogenetics at the NIH. Today, I am going to talk about GWAS for binary and quantitative traits. Going back to the course program, this seminar will be Module 3. Let's get started. I will be walking you through how to run a genome-wide association study, or a GWAS. In recent years, GWASs have increased our understanding of relationships between genotypes and phenotypes. For our purposes, we want to look at variants and their effect on Parkinson's disease. After following modules 1 and 2 on quality control and imputation, we want to show you how easy it is to run your own GWAS. To orient you, here are a few topics we will cover in this module. First, a quick introduction or reminder to what a GWAS is, and important limitations to consider with this type of analysis. Afterwards, I will tell you how to get started and what files you will need. We will go over how to generate a covariates file. More on that later. Next, we will talk about how to run a GWAS in PLINK and discuss other popular tools the community uses, and go over how to make summary statistics and use the popular online tool FUMA. Afterwards, we will go through a few different types of visualizations, what you will need, and what they mean. Towards the end, we will quickly cover what type of regression you want to run based on the data and outcome you desire. First of all, what is a GWAS? A GWAS is a genome-wide association study and a popular method to test common genetic variation associated with a binary outcome such as Parkinson's disease, or a continuous outcome like age at Parkinson's disease onset. This can be done from either exome, genotyping, or whole genome sequencing data, but typically covers all 22 chromosomes. If the variant is significantly found in more cases, meaning patients with Parkinson's disease, versus controls, those variations are said to be associated with Parkinson's disease. The gold standard to identify genome-wide significant associations that are replicable and likely genuine is a p-value of 5 × 10 to the -8. What can we learn from GWASs and why are they valuable? When we find variants that pass at a genome-wide significance, we hope to narrow down where we should start looking for causal effects exerted by nearby genes. While a GWAS can be a very useful tool, it is important to know its limitations. GWASs, by default, only look at common variants, ones found in over 5% of the population, and that are associated with an increase in risk rather than being directly causal. Therefore, it is an inappropriate tool to use if you are interested in exploring rare causal variants with deleterious effects. Perhaps the biggest misunderstanding surrounding GWASs is that the loci found to be associated are in the nearest protein coding gene. However, many studies have proven that the nearest protein coding gene to a GWAS nominated loci is sometimes not the causal gene. Additionally, genetic risk factors associated with an increase in one population might not necessarily hold true for another population. You might find an association in a European population that will not be replicated in an Asian population, since risk alleles and frequencies differ across populations, and this is important to consider moving forward. Since this is a bioinformatics course, we will not discuss these concepts in detail, but instead we'll move on to the practical part. So, what do we need to get started to run our own GWAS? Listed here are the programs and scripts we will need. For this course, we are using R as our preferred language, but you can use any language that you're comfortable with. We will also be using the command line and PLINK. All scripts shown in this course can be found on our GitHub. What else do we need to get started to run our own GWAS? It is important to note that the data we will be using in this course is simulated European data. In Module 1, we were walked through the appropriate quality control, and in Module 2, we were walked through how to impute this data if you were starting with genotype or exome data. Both sets of files will be important moving forward. You will also need a covariate file. More on this in the next slide. A covariate file includes the information found in your PLINK files, like sample ID, their sex, their phenotype, principal components, and all that extra information on samples you might have on hand, like their age, their family history, the cohort that they come from. As long as this is written in a numerical format, it can be included in the analysis. Why is having a covariate file important? By having a file and adding covariates to the analysis, covariates can help explain some of the variance seen in the data and therefore reduce noise. Covariates are coded numerically, and when using PLINK, phenotypes are coded as 1 for controls and 2 for positive cases. Sex is coded as 1 for males and 2 for females. As discussed, you can include a number of things into this file, like sex and age. But how do you calculate the principal components? It is important to note that the principal components are calculated on non-imputed data. The number of principal components you will use in your GWAS depends on how homogeneous your population is. For example, in the Meta 5 paper, the team corrected for 20 principal components, the maximum, even though all the populations were of European ancestry, but this was done to account for variability in variant frequency. You can calculate the principal components in PLINK and then create a covariate file in any language you prefer. For this module, I'll show you how to make one in R. So here is an example of how to calculate your PCs in PLINK. We use high-quality SNPs and narrow down only using common variants, using frequencies at the population level. We prune out unnecessary SNPs and generate PCs on pruned, high-quality SNPs. After we have calculated the PCs, we can now create the covariate file. We read in the eigenvector generated in the PLINK step, the PLINK FAM file, and any additional covariates that you would like to include. Here, we simulated the ages and education level following the FID, IID, and then covariate format, outlined right here. Next, we rename and merge everything accordingly and write out the file. This is an example of what your covariate file will look like, with your FID, IID, paternal-maternal, sex, and phenotype. This is from your plink.fam file. Like I said, we simulated age and education, and these are the principal components that were calculated with PLINK up to 10, and the file will include all of these things merged together. Now that we have all our files ready, can you run a GWAS in PLINK? And the answer is yes. While PLINK is easy to use, it is a bit slower than the popular alternative RV tests. RV test requires slightly different requirements that are outlined in the code on GitHub if you're interested. The typical GWAS is run using logistic regression, and this is because we are predicting a binary outcome. Does the patient have Parkinson's disease? Yes or no. However, you can run a GWAS to predict a continuous outcome, like age at onset. While RV tests require slightly different requirements, in PLINK, however, you can run with the following flags: logistic, covariate name, confidence interval, and hide covariates. We will be using the imputed hard calls generated from Module 2. Using the covariate flag, is how you input that covariates file that we just made, and using the covariate name flag is where we will type out the names of all the covariates we will use. The hide covariate flag is to output the additive effect into our final file, and the confidence interval flag is to output the upper and lower bounds of the 95% confidence interval. Depending on the size of your dataset, this can take a while, but you can see an example of what the output looks like here. Here you can see that we've got the chromosome number, SNP ID, base pair, major allele, number of samples, total used in the analysis, odds ratio, standard error, lower and upper bounds of the confidence interval, and perhaps the most important, the p-value. Here we have created a basic summary statistics file. If you want to explore the frequency of risk alleles in cases versus controls, then at this step, you can run a Fisher's exact test similar to what Hampton covered in the introduction module. Here, we can see the command from the previous slide. It is important to highlight that we only use soft or hard calls post-imputation for a GWAS as outlined in Module 2, ensuring that we use high-quality SNPs. Below you can see an example of the output right here, and you can sort later by the smallest p-value if you're interested. What are summary statistics? Summary statistics, otherwise known as summary stats or SumStats, have aggregate data from cases and controls for each of the variants that have been analyzed and provide p-values to give evidence for the strength of association for each variant. Some other information can be found in summary stats, such as directionality, what the effect allele was, magnitude of effect in beta, which is the per effect allele change in a continuous trait, or odds ratios, which is the exponent of log odds in a binary trait, what the frequency of that variant is. These are just some examples. Summary stats are great because they provide a lot of information without providing individual level data, making it easy to share publicly in most cases. Summary stats, if allowed, should be made public so that other scientists can use that information to inform their own analyses. You can find more summary stats and their associated manuscripts off of the GWAS catalog, sponsored jointly by NHGRI and EBI. Summary stats at this point can be used to put visualizations together or modified slightly to be shared or used with the popular online tool FUMA GWAS, which we will go through towards the end of this module. This is what the GWAS catalog looks like. You can search items of interest. For example, Parkinson's disease. And we can narrow it down by publications as well. As you can see, there's a number of things here. We will revisit this later in this module. Now for the visualizations. As mentioned at the beginning of the module, we will be going through what are and how to make scree, QQ, and Manhattan plots. A scree plot is an easy to make and easy to understand visualization that shows you how much each principal component contributes to the amount of variance we see in the data overall. The reason we make a scree plot is a sort of sanity check to see if the first few PCs really do capture most of the variance and begins to plateau after. If this is not what you see, this usually means there's some sort of big systemic issue in the data, or it's pre-processing, or perhaps you just don't have enough data. On the left of the slide, you can see an example of a good scree plot. This is a plot made from the IPDGC samples that were later used for the META5 Parkinson's disease GWAS, published by Mike Knowles and many others on the behalf of IPDGC. As you can see, the first 4 principal components explain about 65% of the variance seen in the data. The principal components being on the x-axis and the percent variance on the y-axis. The higher a point is on the plot, the more variance that can be explained by that component. Now we will move over to the GitHub to show how to make a scree plot of our own in R. First, we read in the eigenvalues from PLINK. This was generated in the PCs step. Next, we update the column names and scale the values so that they can be plotted properly. After running this script, you will generate your own scree plot that looks like this. Another visualization you can do to check on the quality of your data is a quantile-quantile plot, more commonly known as a QQ plot. Without going into the specifics of the statistics, a QQ plot is simply a probability plot that compares an ideal to an observed. The main reason you would make a QQ plot is to see if there's any systemic biases in your data that would explain why the observed data doesn't match up to the ideal. These biases can be from population structure that wasn't accounted for to genotyping artifacts early on in the process and are known as the genomic inflation factor or the lambda. The lambda scales with sample size, so an important thing to calculate and report is a lambda 1000 value, which reports the inflation factor of a bound study if you had exactly 1,000 cases and 1,000 controls. It is particularly important to report this value for imbalanced datasets. For example, if you had 4,000 cases but 40,000 controls, the lambda would be skewed. But the lambda 1000 value will provide a more accurate idea of what is going on with the data when imbalance is accounted for. On the left here, you can see an example of a QQ plot. The x-axis has the expected or ideal values and makes the diagonal line cutting through the plot, and on the y-axis are the observed values. The expected negative log p-values are generated from a normal distribution, and you want to see how many of your values match up to this distribution. Reported below is the lambda 1000 value, and if the lambda is close to 1, it means the data observed is close to what is expected. If the lambda is less than 1, it means that your study is likely underpowered. However, if your lambda is greater than 1, the inflation could be because of the data or that the study has inconsistencies. QQ plots can be generated before or after imputation, but it is important to note that after imputation, given the number of SNPs, your data is most likely going to be following a normal distribution and will look much better. On the GitHub, we will show you how to generate this sort of plot in R on pre-imputed data. Here you can see that generating an assoc file in PLINK is straightforward, and all you need is the assoc flag and you're good to go. Makes the association file. Once that file has been generated, you will need to read in your PLINK FAM file and this newly made association file. Without going into specifics, you generate the lambda at this step and output your lambda 1000 at this step. This will generate a separate text file with your value and will also export out your QQ plots in these formats, a PDF and a JPEG. Your lambda statistics will be in this lambda_stats.txt file, and your QQ plot will look like this. Perhaps the most iconic visualization to come out of a GWAS is the famous Manhattan plot. The Manhattan plot is named after the Manhattan skyline in New York City, New York, because of its many peaks. It is a type of scatter plot where each plot is a SNP, and the higher the SNP is on the plot, the more associated that SNP is said to be with the phenotype of interest. We make these sorts of plots not only because they're aesthetically pleasing, but because the plot presents hundreds of thousands of SNPs in an easy-to-read way. Most of these plots would include a line to show the genome-wide significance threshold at 5 × 10⁻⁸ making it easy to see just how significant a SNP is. This is the Manhattan plot from Mike Knowles and IPDGC's Meta-5 Parkinson's disease GWAS manuscript. Instead of a line to signify the threshold, they opted to color code genome-wide significant SNPs, with the nearest protein coding gene annotated. Each SNP here is a point, separated by chromosome on the x-axis. On the y-axis is a negative log transformation of the p-values from the summary statistics, similar to the ones that we generated earlier. The smaller the p-value, the higher it is on the plot, and the more significant the association is to Parkinson's disease. While the next step will show you how to use a web platform to generate this sort of plot, we will show you how to make this sort of plot in R. This is based off of the same code used here to make the plot. This particular script requires 2 separate files. The formats of these files are outlined here at the very top of the script. You will need an abbreviated GWAS file containing GWAS summary statistics. Mandatory columns to include are SNP, chromosome number, base pair, and p-value. A hits file is required as well, and this is a file containing annotation for GWAS hits of interest. Mandatory columns to include are SNP, status, and gene. The SNPs of interest are listed with a status indicator, binary, and a gene label, character strings. The script here has a few built-in checks to ensure that you are only visualizing promising candidates. At the end, your Manhattan plot should look quite similar to this. FUMA GWAS is, as they write on their website, a platform that can be used to annotate, prioritize, visualize, and interpret GWAS results. By using the SNP to gene function, FUMA will take in your summary stats as input, though it has to be formatted a particular way, and will return to you GWAS-based and gene-based Manhattan and QQ plots, like the ones that we discussed earlier. They also show some preliminary tissue expression analysis and gene set analysis if you're interested. You can look at GWAS-based and gene-based Manhattan and QQ plots, similar to the ones covered earlier in this module. A few other things that they look at and report are gene set and tissue expression analyses. Let's have a look at their website. In order to get started with FUMA, you will need to register with an email address on their website. The registration button is at the top right. They also require a modified summary statistics file, which is outlined on the GitHub. For this example, we are using publicly available summary statistics published by Blauendrecht and colleagues. This is available on the GWAS catalog mentioned earlier in this module, and is also straightforward to download. There is a link also included for convenience. This is what the entry of these public summary statistics look like. Given that this is a GWAS on age at onset, the authors were assessing a continuous trait, different from the example we have been working through in this module so far. However, to upload these to FUMA, we will need to modify their summary statistics a little bit. We will go over how to do this on GitHub. Outlined in FUMA's tutorial, linked here, they require the file to be in a specific format. This is the code written in R to rename the columns appropriately, but also how to merge the haplotype reference consortium file, linked here, to extract the RSIDs. After following these steps, the output will look like the following. After submitting our file into SNP2Gene on FUMA, the results will take a couple hours to come back. An example of these results using the Parkinson's disease age at onset GWAS on FUMA is linked on the GitHub and looks like the following. A link is also included in the slides for convenience. As you can see, there are Manhattan plots, QQ plots, and other things also reported. As mentioned earlier, typically a GWAS is done using logistic regression. This is because, typically, GWASs are trying to predict a binary outcome. Does this person have the disease? Yes or no? When predicting a binary outcome, we use logistic regression, like the example we worked through in this module. However, you can have a GWAS that looks at a continuous outcome, where there can be many options, like age at onset. If this is the case, you would use linear regression instead. Of course, this depends on your data and what you're interested in analyzing. For continuous traits, the betas are reported, which report the per allele effect. For binary traits, the odds ratios are reported, which are the exponents of the log odds. Continuous outcomes require a slightly modified quality control pipeline. In conclusion, to wrap up this module, some important messages to remember is that GWASs are meant to nominate loci, not particular genes, that pass a genome-wide threshold in the disease phenotype versus healthy controls. In order to do this, however, it is essential that the data that goes into a GWAS is clean and high quality and imputed if they are not being generated from whole genome sequencing. By adjusting for covariates and principal components, you can reduce the amount of noise in your analysis. While incredibly useful, it is important to remember that GWASs also have their limitations. Their biggest one is that they are geared to analyzing only common variants. Finally, if predicting for a binary outcome, like if the person has the disease, logistic regression is used. But if predicting for a continuous outcome, linear regression is used. Thank you very much for your attention, and I hope you've enjoyed this module.